I would like to ask Rana to start. It does not mean that she gets the first prize, so this is this I want to keep closed. And Rana, can you share the screen?
Yeah, give me a second. Hi.
So, and as you know, you have 5 minutes to explain what is on your poster. Yeah, use your poster or slides or what you have, and then we may ask some questions.
All right. So good afternoon, everyone. Thank you for voting for me. So I'm gonna go ahead and start immediately. My poster is titled Gene Associations Based on the Burden of Rare Deleterious Variants.
Anna, can you remove the panel from the speakers? It's like a blank. Yeah. Make it small.
Yeah.
Is this better?
No, not yet.
Give it a few seconds.
Yeah.
Can you see it now?
It's not yet full screen.
Maybe this is difficult because the poster is very large, so she has to focus. Parts, right?
Hold on. Yeah, maybe I need to share my whole screen and then. Is this working now?
No.
So you can't see the poster at all?
Well, we can see the poster, but it's not in full screen. So
yeah.
And just go to full screen mode.
This one?
Yeah, should work.
I know it.
Yeah.
Okay.
Now it's better. Now it's almost perfect. I think we can keep that.
Okay. I mean, I was going to scroll down and up for people to see everything, but I think this should be fine. Okay. So, like I said, my poster Gene Associations Based on the Burden of Rare Deleterious Variants. So basically, we all know there are two basic or usual modes of inheritance. We have either monogenic contribution, where these traits are characterized by low population prevalence, and we usually, in these cases, look for disease-causing mutations. On the other hand, there is the polygenic contribution, in which this phenotype is characterized by high population prevalence, and usually these are common traits that don't have one specific gene that contributes to this trait. However, recently it has been suggested that these two classifications are not enough to, or they oversimplify the genetic contributions of phenotypes. For example, there are common diseases that can be characterized by both monogenic forms and polygenic forms, like diabetes or neurodegenerative disorders or cardiovascular diseases. And also there's a recent work that showed polygenic effects can influence the phenotypic variants and symptoms of monogenic diseases as well. So our objective is to identify gene associations based on an enrichment in rare damaging variants in exome data. On the right side, you can see our pipeline, and you can also see the GitHub page in which you can go and access our pipeline. We basically start with an annotated VCF. We use this VCF anno pipeline, which you can also access. And this annotated VCF contains deleteriousness scores and allele frequencies. We use the equation below. I hope you can see it all. If not, you can visit the poster later in the e-gallery. So basically, this model is multiplicative at a single locus, but additive at the gene level. We multiply by the deleterious score for each variant, and we upweight the allele frequency using two schemas, minus log 10 or beta distribution schemas. And this basically just upweights the variance. So the rarer the variant, the higher the weight of the variant itself. And then after we calculate, we calculate the weights, we make sure that we only select rare variants, so the minor allele frequency less than 1%. We multiply by the deleterious scores. Then we use Blink to incorporate sample-wise scores. And then finally, we output the full scoring. We use association analysis. So after the scores are done, we can do the association analysis. We use three different association analyses schemas. We use Mann-Whitney U-test, logistic regression, and beta-reg. Now, logistic regression, we couldn't use it well because the gene-based scores are not normally distributed, so we can't rely on the logistic regression. That's why we implemented Mann-Whitney U-test as an alternative. However, in Mann-Whitney U-test, we cannot accommodate for covariance. Like, for example, we can't add sex and age. So we decided to use beta-reg as an alternative for that. After that, we can visualize our results. For results, we applied the pipeline on UK Biobank cohort, filtering for British and male samples. So we had about 20K exome data. We used beta distribution weights, and then we done the association analysis on baldness phenotype using Mann-Whitney U-test. As you can see in the results, we had high association of or significant gene in which is AR, androgen receptor, which is also known to be associated with baldness. So in conclusion, it is suggested that complex trait can be influenced by both common and rare variants. We have developed this tool, which can calculate gene-based scores for rare variants to identify gene associations in complex traits. This association analysis that we've shown here identifies AR as significant gene for baldness phenotype, and we hope that we can later use the system to develop prediction models. And thank you for listening.
Thank you for the presentation. So are there questions from the audience? I can't see any. So I have a question. You know this discussion about the missing heritability, I guess. And
Yes.
How much of this can be discovered now with your new approach?
Well, that's what we're trying to see. It's one of the reasons why we're doing this pipeline. We're trying to see if maybe the missing heritability lies in this between monogenic and polygenic contributions. So as I say, I think it's called now oligenic contribution somehow. So it could be that when we try to use these scores along with polygenic scores, we might have some link to the missing heritability that's going on. Yeah.
Okay, thank you. Any further questions?